Hmm. Okay, can somebody confirm you can hear me? I didn't hear that. Let me see if my volume's really low. It's kind of low. All right, I'm going to assume you can hear me because I heard something, but I wasn't sure what I heard. So, um, I'm going to share my screen here. Sorry for being late. I had trouble signing in. Optimize for video. All right, you should be seeing my screen now. Before we begin, are there any questions? I'm not sure we're on chapter 6. Let me see that. I don't know if we finished chapter 5 or not. Let's go to chapter 6 just in case. Let's go back. Yeah, I've got that we were on slide 68. All right. I didn't hear anything, so I'm going to assume there's no questions. Today is May 13th. We are supposed to be on chapter 6, but we need to finish up chapter 5 first. There is no quiz this week. There is a lab today, and sometime tonight, I should be sending you the first unknown experiments and the results to the first experiments, as well as your unknown number. Any question about any of that? Let me remind you, your infectious disease paper is due this Saturday, May 17th. All right, let's begin. So I have that we had talked about metabolic pathways last time. And that most molecules, at least in aerobes and well, anaerobes too, that are using carbohydrate catabolism to make energy, store most of that energy as ATP. There is an integration of metabolism where the anabolic and catabolic pathways are actually joined by common intermediates. And I haven't talked about this. I've talked about anabolism and catabolism being separate and independent reactions. That's not entirely true. They can be connected by an intermediate. We call these pathways amphibolic pathways, metabolic pathways that can function in both directions. They actually bridge the pathway involved in carbohydrate, lipid, and protein metabolism, as well as nucleic acid metabolism. And you all know this. If you were to eat too much carbohydrates, what are you going to store it as? You're not going to store it as a carb in your liver, although you can store some of it, but what are you going to store it as?
It's going to be stored as fat.
Yeah, the carbohydrates that you eat will be converted into lipid and then stored as fat. And that's true of any food material. If you eat too much protein, it would be stored as fat too. All right, let me see if I can blow this slide up. So you can see saloacetic acid goes this way in the Krebs cycle, but it can also go this way. That's why this is a double arrow here. And malic acid can make formic acid, etc. And then we can go to citric acid and actually make acetyl coenzyme A, making acetyl coenzyme A going backwards to pyruvic acid going backwards to glyceride 3 phosphate. And this is being a three-way pathway, why it's drawn this way. We can go from glyceride 3-phosphate back to fructose 1,6-phosphate, and then glucose 6-phosphate back to glucose. And you all know that when you eat too much carbohydrate, it will be broken down largely as glucose, converted to glucose. And it won't be broken down by the Krebs cycle and aerobic respiration. It'll actually broken down in acetyl coenzyme A and whatever that name is, and converted to glycerol, converted to fatty acids, converted into lipids, and we'll store that as fat. The point is that even animals, which are lousy at amphibolic pathways, where reactions can go both ways, there is a common intermediate between carbohydrate catabolism or anabolism, I guess, depending on which way we're going, and protein metabolism, and then lipid metabolism. For lipids, as we mentioned, they would be broken down as glycerol and broken down as fatty acid. Fatty acids will be converted into two carbon chunks and converted into acetyl coenzyme A, adding the Krebs cycle here. But like I said, we can go backwards. We can also go backwards this way, going up to here. And then this one can, glyceride 3-phosphate can make dihydroxyacetone phosphate and then made into glycerol and made into lipids. Any question about any of that? Excuse me here. And I did mention that most of us couch potatoes, I'm certainly a couch potato, except when I'm going out for a walk, it gets a little exercise, but um, most of us couch potatoes, if we were to eat a bunch of lipids and a bunch of protein, meaning a low carbohydrate diet, the lipids over here, and the amino acids will be broken down into glycerol and fatty acids for lipids. And then the acetyl coenzyme A won't go this way because we don't need to make ATP at the moment. We're a couch potato. And the glycerol will be broken down to glycerol, and then we'll join the there's glycolysis at this step, and then they'll go backwards to make glucose. And then glucose will be burnt in aerobic respiration the normal way. So that most of us couch potatoes, we don't burn lipids directly. The lipids will be converted into glucose. And then glucose will be sent down the aerobic respiration pathway when we need ATP, so when we need to burn the glucose. And the same thing with amino acids. The proteins, they'll be broken down into amino acids. Depending on which amino acid they are, they can enter the glycolysis at different steps and the Krebs cycle at different steps. Get even broken down to this amino acid. If you're a couch potato, you're going to make glucose. If you don't need the energy right away to make ATP, and we don't store it as fat, it'll be converted into glucose. And then when we need ATP, it'll be sent down aerobic respiration from glucose in the normal way. Any question about any of that? All right. Let's briefly talk about photosynthesis. Photosynthesis is the conversion of light energy to chemical energy to make ATP. Chemical energy may be used to convert atmospheric carbon dioxide to glucose. We call that carbon fixation. And in that process, the ATP and the NADH that is made by photosynthesis is used up by photosynthesis to make glucose. Photosynthesis is performed by cyanobacteria, algae, and everybody knows green plants. Any question about any of that? Let me briefly state that not all plants are green. Some plants do not engage in photosynthesis, And these are actually parasitic plants that or parasitize green plants, and they are not a green plant and they can't perform photosynthesis. So let me briefly just state that. But most plants are green plants and they do engage in photosynthesis. Photosynthesis is a mechanism for synthesizing organic compounds where you can start with carbon dioxide and water and make glucose. And photosynthesis is required for all life on the planet, with one exception. Uh, and animals and microbes feed on the matter obtained from uh, photosynthetic organisms. There is one exception, and that is the deep sea vents of the ocean. They do not get their glucose being fixed from photosynthesis because they're down too deep in the ocean. Light doesn't travel that deep. The deep sea vents are actually fed by chemosynthetic organisms, which we're not going to talk about, but it's similar to photosynthesis, and that is they fix carbon, CO2, into glucose by using uh, chemosynthesis. They strip electrons from a chemical. But most life on the planet is dependent on photosynthesis to get their glucose. And that includes obviously plants and bacteria and animals too. Even if you eat beef and you don't eat any plants, the cow has to eat the plants. Any question about any of that? All right, photosynthesis only occurs in photosynthetic organisms. These organisms contain a light-trapping pigment. One such pigment is chlorophyll, and it gives the green plant its green color. There are other photosynthetic light-trapping pigments, and as you know, red algae and brown algae have a red pigment that's photosynthetic and a brown pigment, and that's why we call them red algae, because the red pigment gives the organism its color. Anyways, in photosynthesis, light releases electrons from chlorophyll. The light energy is then converted to chemical energy via an electron transport chain. And ATP and NADPH are always generated, but ATP and NADPH may be used to make glucose, in which case they'll be broken down to make the glucose. And glucose will be made from CO2 and water. And we call that carbon fixation. Any question about any of that? Let me mention that NADPH is similar to NADH, but in photosynthesis, they don't use NADH, they use NADPH. There are actually two types of photosynthetic reactions. There's a cyclic photosynthesis or cyclic photosynthetic reaction and a non-cyclic photosynthetic reaction. You've all been taught the non-cyclic photosynthetic reaction, which in the oxygenic phototrophs, meaning those that produce oxygen, this chemical reaction, you need to know this one, is six carbon dioxide molecules get together with 12 water molecules in the presence of light to make glucose, C6H12O6, and six oxygen molecules and six water molecules. Sometimes they'll subtract six water molecules on the right and then subtract six water molecules on the left. I prefer this equation because all of the oxygen generated by photosynthesis, meaning all 12 of the oxygen atoms and the six oxygen molecules, come from the atoms of oxygen. I mean, the atoms of water. So all of these 12 atoms of oxygen come from the atoms of water. And you actually know that from aerobic respiration, where I told you that all of the carbon that we breathe out comes from the burning of glucose. Any question about any of that? All right. There is another non-cyclic photosynthetic reaction involving hydrogen sulfide gas instead of water, and that also fixes carbon and makes glucose. It does not generate oxygen, it generates sulfur. You don't really need to know this reaction. I won't quiz you on it, and other than mentioning it, I'm not going to mention it further. But let me state that there is a cyclic photosynthetic reaction, in which case light does not make glucose. Light only makes ATP. So in cyclic photosynthesis, the chemical reaction is ADP and phosphate get together to make ATP. Here we're looking at the cyclic photosynthetic reaction in a green plant. I know that because this is a membrane of a chloroplast. That's the inner membrane of the chloroplast. But it can happen in cyclic photophosphorylation, can happen in the membrane of a bacteria, in which case it's the cell membrane of the bacteria. Anyways, light strikes the photosystem 1, which is where the chlorophyll is. The light is absorbed by the chlorophyll molecule, which is absorbed by the electron. The electron then leaves the chlorophyll molecule and then moves to this molecule in a chain of proteins in a membrane, to this molecule in a chain of proteins in a membrane, to this molecule in a chain of proteins in a membrane. And then it moves back to this protein, which has the chlorophyll molecule in it, and that electron actually returns to the chlorophyll molecule. Why it's called cyclic photophosphorylation, the electron moves in a circle, returning from where it started from. Along the way, while the electron is moving through this protein, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. That creates a twofold gradient. There's more hydrogen on this side of the membrane than on this side. There's more positive charges on this side of the membrane than this side of the membrane. These hydrogen ions are, by the laws of the second law of thermodynamics, going to want to move from this side to that side. And they can only do that through this channel protein, which is an ATP synthase protein. And when the hydrogen ions move through this channel protein, they give the channel protein the energy it needs to synthesize ADP and phosphate into ATP. This should be familiar to you because this electron transport chain, which is really what it is very similar to the electron transport chain of aerobic respiration right here. It's a little simpler, fewer proteins involved in the membrane, but there's a membrane, there's proteins. The electron moves through the proteins, and then the electrons give certain of the proteins the energy they need to pump hydrogen ions from this side of the membrane to that side of the membrane. And these hydrogen ions can then move through the membrane through this ATP synthase protein to make ATP. Any question about any of that? Excuse me. All right. So the hydrogen ions in a chloroplast are pumped from the matrix of the chloroplast through the inner chloroplast membrane to the space. Oops, pumping it this way, sorry. Pumped from this side of the membrane of the matrix of the chloroplast to this side, which is between the inner membrane and the outer membrane of the chloroplast. In prokaryotic cells, this happens in the cell membrane, so this is inside the cell, inside the cytoplasm. This is outside the cell, so this is actually they're pumping the hydrogen ions into the cell wall if the cell has a cell wall. Any question about any of that? All right. So some requirements of ATP production. This is a summary slide. The energy source can vary. If there's an electron donor, the electrons may occur from sun hitting the photosynthetic pigments, like chlorophyll, causing the electron to leave the chlorophyll. Or the electrons can come from glucose, and then these electrons will be carried by electron carrier molecules. For aerobic respiration, we mentioned that the two electron carrier molecules are NAD plus and FAD. For photosynthetic organisms, the electron carrier is NADP plus. You don't need to know this one. The final electron acceptor can also vary. This graph doesn't show it, but the final electron acceptor of cyclic photosynthesis is the chlorophyll molecule that the electron came from. I don't know why it's not shown here because it has the electron donor varying and the electron acceptor is varying. Anyways, the final electron acceptor of a photosynthetic organism that's engaging in cyclic photosynthesis is the chlorophyll molecule. And the final electron acceptor can vary in respiration as well. For aerobic respiration, the final electron acceptor is oxygen, the last molecule to accept the electrons flowing from glucose. For anaerobic respiration, the final electron acceptor is a molecule other than oxygen. It'll depend on which form of anaerobic respiration it is, but it could be nitrate or sulfate or hydrogen sulfide gas. There's a lot of different anaerobic respirations, and they each have a different final electron acceptor. And then in fermentation, the final electron acceptor is an organic chemical compound, such as pyruvate. Any question about any of this? All right. That's it for chapter 5. Any questions about chapter 5? All right, let's move on to chapter 6. Chapter 6 is going to cover microbial growth. Know the terms of this chapter. What is a psychrophile? What is a psychotroph? What is a mesophile? A thermophile, a hypothermophile, acetophile, halophiles. What's an aerotolerant anaerobe? Facultative anaerobes, binary fission. What are reproductive spores and what are endospores? 2, understand the various requirements of cells for growth. 3, understand the phases of growth. And 4, compare the different methods we have for measuring microbial growth. Remember that when we're talking about microbial growth, we're referring to an increase in the bacterial cell numbers. That differs when you're talking about
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
When we're talking about growth in children, we don't refer to an increase in their numbers. We only refer to their increase in their size. But with bacteria, we refer to growth as the increase in the number of cells. There are requirements for microbial growth, and this is actually true for all growth of any organism, but there are physical requirements meaning the microbes have to grow in the correct temperature, the correct pH, the correct osmotic pressure, the correct salinity. And there are chemical requirements for growth. All microbes have to have a carbon source, a nitrogen source, a sulfur source, a phosphorus source. They have to have a source of trace elements and oxygen if they require oxygen, and organic growth factors if they require those. Any question about that? All right, each species has its minimum temperature, a maximum temperature, and the optimum temperature that it'll grow at. Microorganisms can be classified on the basis of their preferred temperature range. Oops, sorry about that. So here we're looking at the temperature for the physical requirement. Let me blow that up. We have psychrophiles, and they grow best around 10 degrees Celsius. That's the temperature of your refrigerator. And they go down to about a negative 10 degrees Celsius because they have antifreeze. And then they can grow up to about 20 degrees Celsius. But after 20 degrees Celsius, they do not grow. Some of them, if they're very sensitive, they won't grow much above 10 degrees Celsius, but some of them can grow up to about 20 degrees Celsius. The psychotrophs are the organisms that grow best at around room temperature. These will be organisms we find in the soil, maybe in the ocean. Mesophiles are organisms that grow best around 37 degrees Celsius. Remember, 37 is 98.6 degrees Fahrenheit. All human pathogens are mesophiles because they grow best at 37 degrees. Thermophiles are organisms that grow best at 60 to 62 degrees Celsius. And then the hypothermophiles are organisms that they didn't even know when I took microbiology. These are organisms that grow best around 95 degrees Celsius. Recall that 100 degrees Celsius is the temperature the water boils at, and I used to know this, the highest microbe that we have for, that lives, can actually survive a temperature, I think it was like 116 degrees Celsius. Survive and grow up to 116 degrees Celsius. Now, let me state that the exact optimum will depend on the species. For example, let's see, Helicobacter pylori, its optimal growth temperature will be 37 degrees Celsius, so it'll grow fastest at 37 degrees Celsius, but it can grow at other temperatures, but at a slower rate. But my point is we have another mesophile, Micrococcus luteus, that grows on the skin, and it doesn't have its optimum at 37 because the skin is cooler. The skin is closer to around 30 degrees Celsius, and Micrococcus luteus grows best around 30 degrees Celsius, which is the temperature of the skin. So the point is that these classifications are not rigidly defined. The optimum can vary a little bit, and the range can really vary depending on what the organism is. Like Staphylococcus aureus can grow close to about 10 degrees Celsius, but E. coli, well, E. coli's, yeah, E. coli would be a good one, would stop growing like around 20 degrees Celsius. So the range is especially not defined. And then the optimum temperature also depend on the species. So these are not rigidly defined. Any questions about any of that? All right. When we're talking about temperature, it's important to keep food at a correct temperature. Between the degrees of Celsius, 60 degrees Fahrenheit and about 130 degrees Fahrenheit, or 15 degrees Celsius to about 52 degrees Celsius, bacteria can grow very rapidly in the food. And some of the bacteria can produce toxins. And that's why food that has been left out for over 4 hours should probably be eaten promptly or frozen or tossed away. And that's because bacteria can grow rapidly in this temperature range. It can grow slower at another temperature range between 52 degrees and 62 degrees Celsius, or between about 5 degrees Celsius to 15 degrees Celsius. Remember, that's the temperature, well, 10 degrees down below is the temperature of your refrigerator, 10 degrees to 4 degrees. And so bacteria can grow at this temperature. They're going to grow slowly, but after 1 week of growth in the refrigerator, the bacteria can get to numbers to uh, spoil the food. And then let me go back. Remember that the psychrophiles grow best around 10 degrees Fahrenheit. These organisms can grow in your refrigerator. And that's why food that has been in your refrigerator for 1 week should probably be thrown away, unless the food has another preservative in it, like acids or salt or sugar. You should probably throw the food away because the psychrophiles can grow in your refrigerator and they can cause spoilage of your food. The psychrophiles cannot infect you and cause harm to you because if you were to eat them, they are not going to be able to survive at 37 degrees. They'll fall apart, their enzymes will fall apart, their cell membranes may even fall apart. So psychrophiles cannot infect you and cause disease. The only organisms that can infect you and cause disease really are the mesophiles. All right, any question about food spoilage? So the psychotrophs and the mesophiles can grow in food, even in the refrigerator, and that's why after about a week in the refrigerator, they should be thrown away. Unless there's another preservative in the food, like ketchup has both high salt, high sugar, and then high acid. So a bottle of ketchup can stay in your refrigerator for at least 2 years. Actually, I've had a bottle of ketchup in my refrigerator for, gosh, how long has it been? About close to 5 years. And I finally used it up. I don't eat much ketchup, as you can tell. Anyways, food spoilage by mesophiles can happen if, let me blow this up. If you have a large batch of food and then put it in the refrigerator, it can be in the danger zone, 42 degrees Celsius to 15 degrees Celsius, for too long. What is that? 1, 2, 3, 4, 5 hours or longer, which would allow the bacteria to grow long enough to produce a toxin. Bacillus cereus is particularly a bacteria that can grow in rice, and Bacillus cereus produces endospore. So when you cook the rice, the endospore will be surviving. And then if you refrigerate the rice in a too large of a batch, it will go through this temperature range too slowly, and the bacteria can grow and then form a toxin. What you need to do is separate the rice into a smaller batch, and then it can cool quicker in the refrigerator, and then the bacteria do not grow to a large enough number to produce enough toxin to make you sick. They do maybe produce a little bit of toxin, but not enough to be make anyone sick, and they may not even produce enough toxin, any toxin at this range, because they don't have much time to grow. Any question about any of that? All right. Another requirement of organisms to grow is the pH. All organisms have an optimal pH. Most bacteria grow well between pH 6.5 and 7.5, but there are some bacteria that can grow at a lower temperature. We call these acetophiles, and they grow best in a pH between pH 0 and pH 3. And this would be like in a volcanic environment, high acids. Another bacteria that is an acetophile is Helicobacter pylori. It grows in your stomach, and the stomach pH drops to about pH 1.5. If you have certain foods in your stomach, it won't drop that low, but if you don't have any buffers in the food you eat, the pH will drop to about 1.5. Molds and yeast actually grow better, not around pH 7, but between pH 5 and pH 6. In lab cultures, we have a problem when we grow a bacteria because the microbial growth can produce acids which actually inhibit the growth of the bacteria itself. To minimize this acidic buildup, we use pH buffers in our culture media to keep the pH at an optimal pH for the organism to grow. Any question about any of that? All right. Organisms do have a requirement on osmotic pressure. Microbes do require water. High osmotic pressure can remove necessary water from a cell. In a hypertonic environment where the solute concentration is higher outside the cell than inside the cell, meaning you have high salt or sugar out here, the water will leave the cell, resulting in the cell membrane pulling away from the cell wall and shrinking, causing plasmolysis. This can inhibit growth and can even kill cells. E. coli, for example, are extremely susceptible to plasmolysis. And we can use high osmotic pressure to preserve foods like salted meat, salted fish, and molasses. When the bacteria fall in the bottle of molasses, the water will be pulled out of the bacteria, and that will kill many organisms. E. coli would be dead fairly soon, like within a day or so. Some bacteria, like Staphylococcus aureus, could survive for longer than a day, but given about a week, it would die too, maybe even faster. Endospores would not die, but they could not germinate in the bottle of molasses. And that's why molasses can stay in your cupboard at room temperature for years. My mother had a bottle of molasses that we figured was over 30 years old. And she finally said, you know, you're not using this molasses. I'm not using it. I'm going to throw it away. Any question about any of that? And we know the molasses were good at 20 years old or older because I made my father some molasses candy and he ate it and didn't suffer anything from eating it. Halophiles are organisms that like high osmotic pressure and they like it for coming from salt. There are obligate halophiles They require a high salt concentration to grow. And then there are facultative halophiles. They do not require a high salt concentration to grow, but they can tolerate it, and they can grow at salt concentrations higher than 2%, growing at salt concentration at 15% or higher. Staphylococcus aureus is a facultative halophile. It can tolerate salt concentrations of getting up to 20% sodium chloride. That is the highest salt concentration that your skin can get when you don't take a shower or bath for a long period of time and you've been sweating. But they don't require high salt concentration to grow. They don't even require 2% sodium chloride. They do require some, but they can grow in 0.9% sodium chloride, which is isotonic to our cells. It's also isotonic in most bacteria. An obligate halophile would be one that requires high salt concentration to grow, and some of the bacteria growing in the Dead Sea between Jordan and Palestine, they are obligate halophiles. They can only live and grow in high salt concentration. If you don't know, the Dead Sea has about the highest salt concentration anywhere in the world, about 30% sodium chloride, which is as high as you can get sodium chloride in water. In a hypotonic solution, low osmotic pressure, meaning we have lots of water around, water will enter the cell by osmosis. If the cell does not have a cell wall, it has to be a complete cell wall, then the cell will lyse. You put red blood cells, which lack a cell wall, in a beaker of water, water will flow into the red blood cell, causing the cell membrane to expand. More water will flow into the cell, causing the cell membrane to expand further until the cell membrane pops, and then that will kill the cell. Any question about any of that? All right. There are chemical requirements of organisms to grow. All life forms must have a carbon source. Carbon is typically half of the dry weight of a cell. Doesn't matter what cell we're talking about half the cell's weight is carbon. Carbon is used in structural and organic molecules, like the structure of your membrane. At least there's some carbon in that, both in the lipids and the carbohydrates. Carbon is also an energy source for organisms. Heterotrophs use an organic carbon source. We are a heterotroph, and we get a lot of our carbon source from glucose and the other food we eat. In fact, we get all of our carbon source from that. Autotrophs, on the other hand, can get their carbon source from CO2 dissolved in the air or the water. Any question about any of that? Nitrogen, sulfur, and phosphorus are three other chemical requirements that all organisms require. Nitrogen, about 14% of the dry weight of a cell. Nitrogen is found in amino acids, in protein, DNA, RNA, and ATP. Nitrogen is often obtained by microbes from the decomposition of protein. But many bacteria can use ammonium ion or nitrate ion as their nitrogen source. Green plants can too. In fact, nitrate is one of the main ingredients in fertilizer for plants. A few bacteria can even use nitrogen from the air, gaseous nitrogen, and then fix it, pulling down the nitrogen in the air into an organic molecule in the bacteria or archaebacteria. Sulfur can be obtained from amino acids or protein, thiamine, and biotin. Biotin is a B vitamin. Sulfur is required for DNA and RNA synthesis. Sulfur is often obtained from the decomposition of proteins, but many bacteria can get sulfur from sulfate or hydrogen sulfide gas. Hydrogen sulfide gas is a toxin to us. Sulfate is not a toxin, but we can't get our sulfur from sulfate, but green plants can. So sulfate is another ingredient in plant fertilizer. And many bacteria can get their sulfur from a sulfur source other than a protein or amino acid or thiamine or biotin. Phosphorus is used in DNA, RNA, and ATP. Phosphorus is also in the cell membrane, in the phospholipid of the cell membrane. Many organisms get their phosphorus from phosphate, including us. We can get our phosphorus source from phosphate. Any question about any of that? All right, all organisms require trace elements. These are organic elements required by microbes in small amounts. They're often required as an enzyme cofactor. Some examples of a trace element include iron, copper, and zinc. Trace elements are usually picked up from their environment, either in the water or wherever the microbe is, the soil. They can usually get it from their environment. That's true of humans too. We do require copper and zinc, and we usually get those as trace elements in either the food we eat or the liquids we drink. Any question about any of that? Organic growth factors may be required by certain microbes, or an organic growth factors or organic compounds obtained from their environment that the organism cannot synthesize itself. Some examples of organic growth factors include vitamins, certain essential amino acids, certain essential fatty acids, and purines and pyrimidines. An organic growth factor for humans is vitamin C. In fact, all of the vitamins, except for maybe vitamin A and D. Is it D? Yeah, D. We can actually make vitamin D when we get the precursor and sunlight, but all the other vitamins we have to get in our diet, like vitamin C. Any question about any of that? An example of an organic growth factor required by a microbe is Streptococcus pyogenes. Needs something that red blood cells require. I think it's NADH, but it's the factor V in the red blood cells. My point is that Streptococcus pyogenes does require an organic growth factor, and that's why Streptococcus pyogenes cannot grow on a general purpose media. It needs to grow on blood agar or chocolate agar to supply that organic growth factor for the bacteria, Streptococcus pyogenes. Any question about any of that? All right. Oxygen is required by aerobes. Molecular oxygen is, in a sense, a toxic substance, even to an aerobe like ourselves. We can handle oxygen gas, but there are 4 toxic forms of oxygen, which are toxic to all cells, including our cells and other aerobes. There's singlet oxygen, where the oxygen molecule is boosted to a high energy state, where the electron has such high energy that the oxygen is unstable, and the singlet oxygen is extremely reactive. It's actually present in our phagocytic white blood cells, and when our white blood cell comes up against something it wants to kill, it can secrete this singlet oxygen onto what it wants to kill. Or it can also engulf what it wants to kill and then release the singlet oxygen on the thing engulfed inside it to kill it. Any question about any of that? Superoxide free radicals are another toxic form of oxygen. This is oxygen gas with an extra unpaired electron. That's what that, let me blow that up. What that square stands for, an extra unpaired electron. It has an extra electron, so it has a -1 charge. Any molecule which is a free radical, which has an extra unpaired electron, is highly unstable, and it likes to either lose that electron or gain another electron, because electrons don't like to have one extra. They like to have either 2 extra or no extra. If you have 2 extra, then we have oxygen with a -2, and that's actually stable. But oxygen gas is normally no extra electron and has no charge, and it's also stable. Anyways, superoxide free radicals, that's the oxygen with an extra electron, is made at low numbers in aerobic respiration. Aerobic respiration, remember, is when oxygen is used as the final electron acceptor. This oxygen radical steals an electron from whatever it bumps into. And then that molecule becomes another free radical, which can steal an electron from another molecule. The trouble is that they can steal the electron from DNA, mutating the DNA. Superoxide free radicals can be neutralized by the enzyme superoxide dismutase, more commonly called SOD, in which case we get 2 superoxide free radicals and 2 hydrogen ions in the presence of the enzyme SOD to make hydrogen peroxide in water. But hydrogen peroxide is also toxic to cells, but the cell has a way of handling that. And that's our third toxic form of oxygen, and that's a peroxide anion, such as hydrogen peroxide is a peroxide anion. Our cells can handle hydrogen peroxide or a peroxide anion by the enzyme catalase, converting the hydrogen peroxide into water and oxygen. Hydrogen peroxide being broken down into the oxygen will create bubbles within the hydrogen peroxide. We talked about that in the catalase reaction before. There is another way you can break down the peroxide anions or the hydrogen peroxide, and it's the enzyme peroxidase, converting hydrogen peroxide and 2 hydrogen ions into 2 water molecules. All organisms that live in oxygen have to have either catalase or peroxidase or both of these enzymes, or else they cannot survive in oxygen, because peroxide anions are made in low numbers in aerobic respiration. And the peroxide anions or hydrogen peroxide would be toxic to the cells. And like I said, our cells handle this by catalase. E. coli would handle that by catalase. Members of the Streptococcus family handle that by peroxidase. Like Streptococcus pyogenes handles that by having peroxidase. All right, the fourth toxic form of oxygen is the hydroxyl radical. This is a hydroxide with an extra electron that is unpaired. Normally, OH has a -1, meaning it has 2 electrons extra, and that gives it the -1 charge. The OH with 1 electron is Free radical. Hydroxyl radicals are made by aerobic respiration in trace amounts. They're also made by ionizing radiation. And whenever hydrogen peroxide reacts with certain metals, they make the hydroxyl radical. Our cells get rid of hydroxyl radicals by having antioxidants, which then combine with and then remove the free radical. Vitamin C is actually an antioxidant that combines with hydroxyl radical, removing the hydroxyl radical from our cells. The trouble is that would use up the vitamin C, meaning that when the vitamin C joins with the hydroxyl radical, it removes the hydroxyl radical, but it also removes the vitamin C. Any question about any of that? Vitamin E is another hydroxyl radical removal. Note that the hydroxyl radical is not the hydroxide ion with a -1 that has another electron. It's also not the hydroxide of a hydroxyl group of the OH in alcohol that's connected with an R over here. Any question about the 4 toxic forms of oxygen? Microbes that use oxygen are called aerobes, and they produce more ATP from nutrients than anaerobes that do not use oxygen, meaning they do not have oxygen as the final electron acceptor. Microbes can be classified on the basis of the effect oxygen has on their growth. I'm going to blow this slide up. If the organism grows best and only where there's oxygen, we'll say them growing highest at the top, because that's going to be 20% oxygen. But as you grow down the thioglycolate tube, we'll have less and less oxygen. Thioglycolate is a media that removes oxygen from the media. But once you take the tube out of the autoclave, oxygen from the air will start moving into the thioglycolate. And at the very top of the tube, it'll be full oxygen, 20% or whatever oxygen is. But as you go down the tube, there'll be less and less oxygen. And we'll see less and less growth because the aerobe requires oxygen to grow. At the bottom of the tube, we wouldn't see anything. And in this region, we have just a little bit of growth because there's a little bit of oxygen right here. So as you go up the tube, you'll have more and more growth. I'm trying to make a triangle there. Most growth at the top, less and less growth as you go down the tube, till you get no growth because there's no oxygen. The next easiest to explain is the obligate anaerobe. These organisms only grow in the absence of oxygen, and so at the bottom of the tube, you get the best growth. As you go up the tube, there will be less growth. You got 3 there. Let's put 1 here, because there's a little bit of oxygen there. So we'll have a little bit of growth because there isn't much oxygen. But as you go up the tube further, there'll be no growth because there's too much oxygen. And it's the oxygen toxic radicals or toxic substances that cause the cell, the obligate anaerobe, to die. One of the other easier ones is the facultative anaerobe. They can use aerobic respiration to grow, but if oxygen is not around, they can use some other means to grow, either fermentation or anaerobic respiration. So they can grow in the presence of oxygen, and they can grow in the absence of oxygen. So these bacteria will grow everywhere in the tube, but they'll grow best at the top of the tube. And as we go down the tube, we'll have less and less growth, but we'll have growth all the way down to the bottom of the tube because they can grow without oxygen. Any question about any of that? All right. The next one, which is fairly easy to explain, is the aerotolerant anaerobes. These are anaerobes, They do not require oxygen to grow, but they have all the enzymes they need to remove the toxic forms of oxygen. So they can grow in the presence of oxygen, and they can grow in the absence of oxygen, and the growth will be even throughout the tube because they have the enzymes they need to remove the oxygen toxic molecules from the cells, but they don't need oxygen, so they can grow without oxygen. There's only a few microbes which are aerotolerant anaerobes. And then there's a few microbes which are microaerophiles. These are aerobes. They do require oxygen to grow. However, they do not grow best at full oxygen. They grow best at oxygen levels something between 2 and 10% oxygen. This could be Helicobacter pylori, a microbe that grows in your stomach. You do have some oxygen in your stomach because when you swallow, oxygen will go into your stomach. However, the microbes in your stomach that are facultative anaerobes or aerobes will use up that oxygen, reducing the oxygen in your stomach to less than 20%. And Helicobacter pylori prefers a lower oxygen level to grow, something between 2 and 10%. So these organisms grow best at reduced oxygen levels, but they require oxygen or they don't grow. So as you go down the tube, you'll get less and less growth. To the bottom of the tube, you get no growth. And then as you go up the tube, you'll also get less and less growth because they do grow in the presence of oxygen, but they won't grow as well in full oxygen. So growth occurs someplace in the middle of the tube. Any question about the effect of oxygen on the growth of microbes? If not, let's change gear and we're going to talk a little bit more about media. We've already talked about a general purpose media. This is a medium which, when you have several of them, you call it media. If there's one, it's medium, singular. It is a liquid or a solid solution containing all the nutrients required by microbes to grow. When you get it in the lab, it'll be sterile, meaning containing no living microbes. And how we sterilize it is we put it in the autoclave, which is like a giant pressure cooker. I think we talked about autoclaves already, but in case I didn't, it's sort of like a giant pressure cooker, killing all bacteria, including endospores, as well as all cells. The inoculum is the microbe we introduce into the media. The culture is the microbes growing in or on a culture medium. We have a wide variety of media available for you to grow in a microbiology lab. You see this book, which isn't quite a book, but it's close enough. It's really thick, really big. In the lab, we have a book. It's actually a little bigger than this, which on every page of that book lists one media. So you flip the page, you'll get another media. That tells you how many different forms of media we have available in microbiology. So there's a wide variety of media because this is a large, very thick book, large number of pages. Any question about any of that? To support microbial growth, a media must provide an energy source, a carbon source, sources of nitrogen, sulfur, or phosphorus, trace elements, and organic growth factors if an organism needs these factors. Like Streptococcus pyogenes would require all of these. And that's why we put blood in there to supply the organic growth factors. Trace elements are usually obtained from the media itself, like the agar can supply the trace elements, or the water. A source of nitrogen can be protein in the media. Sulfur, they may include sulfate in the media to supply sulfur, and phosphate in the media to supply phosphorus. Carbon source could be carbon in a carbohydrate like glucose. And the energy source could be supplied by the glucose as well. But what does a photoautotroph organism require in the media? And let's say a phototroph, so we're getting an energy source if we're supplying light, and it's an autotroph. And let's also add in, it fixes nitrogen from the air. So what do we need to supply for this photoautotroph? Do we need to supply an energy source, assuming we're giving it light? It's a yes or no question, people.
Yes.
Thank you. I wasn't going to move until somebody stated. No, actually, we do not need an energy source if we're supplying light because this is a phototroph. And do we need a carbon source? Remember, this is an autotroph. We don't need a carbon source because it can use CO2 from the air or dissolved in the media. We would not need a nitrogen source because it can fix nitrogen from the air. We would need to supply sulfur and phosphorus and trace elements, which usually come from the water. And it's an autotroph, so you don't need any organic growth factors. So all you need is the trace elements from the water, so we don't even need to add them, a source of sulfur, and a source of phosphorus. So in this media, we're not supplying much. But if we're growing E. coli, we have to have an energy source, and that would probably be glucose in the media. We have to have a carbon source, and that would probably be glucose in the media. We have to have a nitrogen source, and that would probably be protein in the media. And we have to have sulfur in there, sulfate, phosphorus in there, phosphate. We have to have supply trace elements, but that will usually come from the media itself or the water. And E. coli doesn't require any organic growth factors. All right, any question about any of that? Agar is a complex polysaccharide derived from seaweed. We use it as a solidifying agent for most solid culture media. You can use another solidifying agent like gelatin or starch, but most of the time we'll use agar. Agar is usually used in a petri dish because having a broth in a petri dish is just too dangerous. You have to hold the petri dish perfectly, or you contaminate the dish by spilling the broth out of the petri dish. We can also use agar in a tube, and we have two types of agars in a tube. We can have a slant where the agar is put in when it's a liquid, and then you put the tube at a, sorry, I went too far there, at an angle, so it's sort of like that. Or we can have the agar in a tube like this, where the agar just goes flat across. Meaning we put the agar in when it's liquid and then it solidifies, making a deep. We call this a deep and that a slant. Any question about that? You can also have a broth in a tube, and we don't really call that anything. We don't call it a deep or anything else, just a tube of agar. Not agar, a tube of broth. Agar is generally not metabolized by microbes, and that's why it's still used today. Initially, Dr. Koch He actually started using solid media, and what he did was he used a potato, which was not a very good substance to grow your microbes on. And I don't know if he used a cooked potato or a raw potato, but he streaked his bacteria on a potato. And that wasn't the best thing to do. So he put, in a petri dish, he put gelatin. The trouble with gelatin is some microbes can digest the gelatin, and then he got this gooey liquid mess in his petri dish. And it was actually the wife of one of his what do you call it, lab technicians who suggested that he try agar. Agar was a substance used for solidifying foods or adding thickness to food like soup. And he did, and he liked it and found that no bacteria could digest it. And we still use agar today to solidify our liquid media to make it a solid. We have different types of culture media. There are chemically defined media where the exact chemical composition of the media is known because all of the molecules are added by you when you make the media. Like this media is a chemically defined media. It's made by putting 5 grams of glucose, 1 gram of ammonium phosphate, 5 grams of sodium chloride, 0.02 grams 0.2 grams, sorry, of magnesium sulfate, and then 1 gram of potassium phosphate in 1 liter of water. These are the only molecules in the media. At least we use distilled water. These will be the only molecules in this media. Chemically defined media. The trouble with the chemically defined media, it's saying my internet connection is unstable, so if you need me to repeat something, please let me know and I'll repeat it. Anything you need repeated? The trouble with the chemically defined media is it will not grow a fastidious organism because a fastidious organism will have certain additives required. Like this is a media. Let's see if I can blow that up. That is a fastidious organism. You have to add a whole bunch of carbon and energy sources like oxaloacetic acid. You have to add a whole bunch of salts, ferrous nitrate, so nitrate and iron. You have to add amino acid like cysteine, arginine. Cysteine? Isn't that cysteine? I don't know what that is. And organic growth factors. Thiamine, that's a B vitamin. Nicotinamide, adenine dinucleotide, NADH. Uracil, biotin, those are all B vitamins. So this is a fastidious organism. We can't get it to grow unless you add all of these additives, which would be difficult to make that type of media. Normally, chemically defined media are only used for autotrophs because they do not require a bunch of additives to the media that need to be added. An autotroph is something that can make its own glucose by fixing CO2. Normally, we use a complex media like one where not all of the molecules are known because we're adding extracts of yeast, meat, or plants, providing lots of vitamins and organic growth factors. The exact chemical composition is not known. Let me go and blow this up. Like this complex media has digestive protein forming peptone, digestive beef, you grind up the cow, sodium chloride, agar, and water. But the exact molecules in the cow will depend on what the cow has been eating, where the cow is living. And so we don't know what all the molecules will be in this media because the cow will supply many. The peptone usually comes from soybeans. Depending where the soybeans grow, it will have different molecules in it as well. We usually use a complex medium for growing heterotrophs because these bacteria require organic carbon source. Any question about any of this? If not, I'm going to stop here and I'll see you at 6:30 in the lab. Any questions? All right, bye.